Okay, so our next topic we are having that is classification of enzyme. Okay, so the classification of enzyme, if we talk about, so we are having the classification on the base of the, we are having reaction type and substrate type. Okay, so but in this video we will discuss about the reaction type. In the next video we will discuss about the substrate type. Okay, so on the basis of the enzyme we are having six groups. Okay, six group. Number one group we are having that is oxidoreductase. What does it mean? So as you guys all know, for before coming on that point, we are, let me give you an point. So what does the oxidation means? Oxidation means the loss of hydrogen or electron or oxygen. If we talk about the reduction, so we are having that a molecule that is gaining this one, these all, we are calling that as a reduction on that of a molecule. Okay, so these basically are the enzyme which basically you can say that add the electron or hydrogen ion from the enzyme to the substrate. Okay, for example we are having the example of the enzyme. We are having cytochrome oxidase. So this basically is the enzyme which basically oxidizes cytochrome, which means that it basically, you can say that this is basically oxidize the cytochrome. Okay, so this is was the oxidoreductase. Now the next group we are having that is transferase. What does it mean? So the transferase, if we talk about, these are basically the enzyme that basically not transfer this one, the hide this one is what, this is the hydrogen electron oxygen. This will not transfer this will transfer the functional group. For example, as you guys all know, in the glycolysis we have discussed about the glucose is converted to glucose 6 phosphate. So here, as you guys all know, we were having the ATP, and ATP was the one phosphate was going to the glucose and formed, and this one become ADP, and so this one was formed as a glucose 6 phosphate. So the enzyme that was used here, that was transferase. Okay, not transferase, this is the process transferase. So enzyme that was used on this step we were having, that was hexokinase. As you guys all know, in the botany video and in the botany lecture we have discussed about the glycolysis. So in that the hexokinase was there. So hexokinase, what this is the transferase. So after that, if we talk about the transferase, so we were having the enzyme, so glucose 6 phosphate. Okay, so the glucose 6 phosphate was converted by the glucose. So the enzyme that was used, that was hexokinase. Okay, so this is basically the functional group that is attaching. Okay, here the oxygen electron hydrogen was doing the same thing. After that we are having the enzyme that is, we are having that is hydrotases. Hydrolases. Okay, so the hydrolases was an enzyme. This was basically a group of enzyme which basically add the water molecule. Okay, and break the product, which means that the addition of a molecule, and for example I'm having this A plus B. Okay, so I'm adding the water H2O. Okay, so the A will bind with the hydrogen and B will bind with the hydroxyl group. Okay, so this is basically what, this both get detached, so this is called the hydrolases. So with the water add, the molecule will get break, that is called the hydrolases. The enzyme, if we talk about, that is pepsin, which work in the stomach. We are having rennet, which work in the stomach. We are having trypsin, which work in the, you can say that the, in the large intestine. So these basically both, these all the enzyme basically are the proteolytic enzyme. This basically break the protein into the protons, peptides. Okay, so these are the enzymes. So these are basically work by the addition of the water and break the molecule. So this was hydrolases. After that we are having another group, that is lyases. What does it mean? It means that the bond will break, but without water. So this was the enzyme that basically break down the specific covalent bond without the hydrolysis. For example, we are having the enzyme that is histidine decarboxylase, which means this is basically the carbon will be removed in the form of the carbon dioxide. Okay, so this is the histidine decarboxylase. This basically break the you can say that the bond by the removal of the carbon in the form of the carbon dioxide. After that we are having that is isomerase. As you guys all know, in you can say that we were having the in the glycolysis cycle, as you guys all know, the glucose was converted into the glucose 6 phosphate. After the glucose was 6 phosphate was converted into the fructose 6 phosphate. So this is the conversion. Here again these are product are same, but this is the you can say the isomerase, like glucose, fructose, galactose, these all were isomerase. So which means that they are having the same formula C6H12O6, but their structure is different. Okay, so this is converted into the fructose 6 phosphate. So the enzyme that was used here, that was isomerase in the glycolysis cycle. So this isomerase basically this is the photohexoisomerase. We are calling as a isomerase. This is the photohexoisomerase, which was work in the glycolysis with the conversion of the 6 molecule of the glucose into the 6 molecule of the fructose. So this was the enzyme that was used. So we are calling that enzyme in the isomerase group. So this was the enzyme. Okay, so which was changed into the glucose 6 phosphate into the fructose 6 phosphate. So this was the isomerase. After that we are having the enzyme group. We are sixth one, last one is the ligase. Ligases. What is ligases? Ligase is basically a molecule is joined with a molecule. With the help of this enzyme, a molecule is joined with another molecule. With the help of the enzyme, we are calling that enzyme as a in the ligases group. For example, we are having polymerase. Polymerase, as you guys all know, we have discussed have you seen in my videos, in the botany videos we are having. So this enzyme I have used many time in the lectures, in the botany lectures. So this was a polymerase enzyme which was basically binding the two molecules. Okay, so this is basically responsible for linking the monomer into the polymer, you can say, as such as DNA we are having, RNA we are having. So this is the enzyme, basically they link two enzyme together and make a product. So this enzyme is like you can say that belong from the ligases group enzyme. So this was the classification of the reactants. Okay, reaction type. So we are having 6 groups. So today we have discussed about the reaction type. In the next video we will discuss about the substrate type.